instruction. A very pleasant morning to all the participants, and on behalf of the St. Xavier's College, Department of Zoology, and the organizing team of the National Level Zoology Webinar Series 2020, we extend a warm welcome from the bottom of our hearts to all the participants who have gathered here on the third day. Before entering the session, I would like to give a few instructions. It is a small appeal from our side. The participants, you can join the webinar by clicking the link provided in your mail or by using the meeting ID and the passcode. Request you all to mute your audio and turn off your video during the session. And you can join the webinar meeting in your name and mention your department and college and type the same in the chat box. If you have any question, please post your question in the chat window during the webinar on your behalf. We will raise your questions to the resource person. And kindly submit your feedback immediately at the end of the session. The link will be active only for 15 minutes. And then you can leave the meeting after filling the feedback form. So if you experience any connection problem, kindly click the link or use the meeting ID and passcode to re-enter into the webinar room. Today we have two sessions. Before entering the session, I would like to give a small introduction. There is gold, there is rubies in abundance, but lips that speak knowledge are a rare jewel. And here we have two eminent personalities to share their knowledge with us today. And we are here, the heart of the discerning. It will seek and acquire knowledge, and the ears of the wise will seek it out. So do not forsake this wisdom, because it is going to protect you and it is going to watch over you. Wisdom is supreme, and therefore we ought to search and obtain wisdom. Though it costs everything you have, we have to obtain understanding. So today for the first session, we have Dr. Febin Prabhudas. He did his B.Pharm in Madras Medical College, M.Tech Bioinformatics in Shanmuga Arts and Science Technology and Research Academy, Tanjavur, and PhD Bioinformatics in Vellore Institute of Technology, Vellore. He was awarded 2 Indian Academy of Sciences Summer Research Fellowship, one at the Center for Cell and Molecular Biology, Hyderabad, and the other one at Bangalore. He has published 24 research articles, 1 book chapter, is presented in 4 international conferences, including visiting countries like Austria, Singapore, and Canada. He has guided 5 doctoral students, and his research includes programming in bioinformatics and molecular evolutionary studies on G protein coupled receptors, HCV infection and clearance genes, drug designing and targets, codon usage in genomes. He's skilled in many bioinformatics tools like PAML, PHYLIP, GROMAX, GCG Wisconsin, MATLAB, MetaTool, CellNet Analyzer, Series 2, Insight, Modeler, and SPDBV. Dr. Febin Prabhudas is at present the Associate Professor in the School of Biosciences and Technology, Vellore Institute of Technology, Vellore, handling bioinformatics course, conducting various research workshops and short-term courses, and actively involved in research. We are honored to have Dr. Febin with us for the first session today. Welcome you, sir. Dr. Febin, can you hear me? Dr. Febin?
Yeah.
Dr. Febin, can you hear me? We welcome you, sir. And you can
Thank you for
start your presentation.
introduction.
The session is all yours. Okay, welcome.
Yeah, thank you. So, everybody can see my PowerPoint?
Yes, sir.
Hello?
Yes, it's visible.
Yes, okay. Okay, so first of all, I thank the St. Xavier's Institute principal and the organizer, Dr. Mabel, Parimala, and the supporting staffs, and the students who are eagerly waiting to see my lecture delivered. So I'm happy today morning to share my little research work which has been done 2 years back along with my PhD student Arti Venkatesh. So today, as you all know, that my topic is computational probing of antiviral drug for HCV. And since I'm basically a bioinformatics teacher, and I've been working here in BIT since 2005, I have ample opportunity to work on different projects. But because it's the bioinformatics is basically a supporting domain or a discipline which could reduce the burden of the biological work or experimental work. Instead of maybe checking, for example, now we take the coronavirus, which has been recently became pandemic, where you can instead of taking millions and millions of compounds and searching for the right drug for this coronavirus. So if we use the bioinformatics application, we can automatically reduce the number of the drugs that we can take it to the next level. So it has wide applications, the bioinformatics. So today I'm going to talk about the antiviral drug for HCV. Okay. So the introduction, what is HCV? The HCV, as we all know, that it's inflammation in liver, the hepatitis. Okay, so all the itis are nothing but the inflammation. Identified in the year 1989 as a non-A and non-B hepatitis, and the serological blood tests have been available since 1992. And the mode of transmission is from unsafe injection practices, inadequate sterilization of medical equipments, transfusion of blood and blood products. And in fact, the World Health Organization have shown the data in 2018 that 7 million deaths have occurred. That is around 3% of the population have been affected with this disease. So no effective vaccine available to prevent this deadly disease till date. So this is the representation of normal liver, and this one is the liver with hepatitis B. So the structure of the hepatitis C virus. So hepatitis C virus is a quasi-species of Flaviviridae family, and the genus Hepacivirus, and it's very much closer to the the corona size is also around 60 nanometer, and it's a single-stranded positive RNA virus. And it has 6 major genotypes, and subtypes are more than 100 based on the country they've been identified. So signs and symptoms include the abdominal pain, confusion, drowsiness, loss of appetite, swelling in the legs, yellow discoloration in urine. Okay, so this is how the virus get entry to the liver cell, and it completely hijacks the machineries of liver cell, and it has been released out of the cell as a new hepatitis C virus. So in fact, we all know that all the viruses, RNA viruses, protease is a very important enzyme which will be used to take the machinery or utilize the machinery of the host cell and produce a new hepatitis C virus. Okay, so the classification of HCV genome. So as we all know that the genome is the complete DNA sequence, or the number of nucleotides present in any organism, even the human genome. Together the entire DNA material of the genome, we call it as genome. And the HCV genome is made up of only 9,600 nucleotide base, and I think HIV virus is composed of 8,200. Okay, roughly around that number. So little larger than the HIV. HCV genomes are classified into structural proteins, non-structural proteins. So under structural proteins we have C, E1 and E2. And under non-structural proteins we have NS1, NS2, 4A, 4B, 5A and 5B. So totally only about 10 proteins, right? Okay, so treatment methodologies are adopted for HCV. In the 1989, it has been identified, and 1991, the first approved alpha interferon, and next one is pegylated interferon in 2001 after 10 years. Again, after 10 years, the drug has been started to come through the Incivex. And 2013, it's been growing. The number of drugs have been started growing. The problem with these all drugs are having severe side effects. Okay, but still the people are trying to find a better drug. So these are the FDA approved drugs for HCV for each and every protein target. So first is the NS3 protein, NS3-4A protease inhibitor, and the NS5A inhibitor. Also nucleoside and non-nucleoside inhibitors. So these are the, for example, bokipravir, sovapravir, asunapravir. Okay, so these are the FDA approved drugs for HCV. So workflow for this work is basically finding, taking all the antiviral drugs which are known to inhibit or used for other antivirus also. I mean, as an antiviral drug. So collecting all, including pooling this also, the HCV drug which have been shown in the last slide also, put everything together and find the molecular properties. Okay, and that's a major idea. So it's nothing but the features of every drug, right? For example, if you take a flower, a rose flower, right? And if I ask what are the features of a rose flower, you will automatically say the color is rose, and you will say that they'll be having some thorns or the petals, the shape, size, length, all those things you will be saying, right? So same way, all the drugs will be having their own molecular properties. There are about 4,900 and some odd physiochemical properties are there. So if I'm giving you two flower, right, a rose and a jasmine, and I'm asking you to find the or three flower I'm giving, okay, find which is closer to rose, jasmine, and hibiscus. So, and I'm asking you to find which looks closer to rose, whether the jasmine or the hibiscus. So, what you will be saying, the hibiscus automatically, because the color and the size, shape, and everything. So, similarly, with the known drug having their own physical properties, physiochemical properties for HCV, and also the other drugs that has been used in some other antiviral treatment. So what we are trying to do is, so rose is nothing but the HCV drug which is already present, and hibiscus and other type of flowers are nothing but the flowers which is going to match the rose or match the HCV drugs, right? Okay, so what we do for that is computation of 2D molecular descriptors is nothing but the physiochemical features, and clustering. What is, okay, clustering is grouping all the flowers or the keys or whatever based on the similarities of the parameters that are close to each other. Parameter similarity in the sense I said, no, the length, breadth, color, whatever features that are closer to the existing one. So next, docking studies is to carry out the binding, energy and interaction between the HCV and non-HCV inhibitors to their respective proteins. So these are the two drug targets that we have taken today. Is the NS3 has been identified with the endoplasmic reticulum that forms heterodimeric complex with NS4A, which is 54 amino acid membrane protein, act as a cofactor of protease. And what the N terminal of this protein does is it is having a serine protease activity. That is, it also it also has the helicase activity. It cuts the DNA sequence. Next is the next protein is the NS5B protein, and which is the C terminal region of the HCV protein. HCV. The main role of the NS5B protein helps in viral replication and it also helps in the synthesis of RNA, which blocks the enzyme activity. So these are the proteins which are present in the virus itself, which try to hijack our machinery through some molecular mechanism. So if we inhibit these proteins, if we find a key for this log, then we can easily stop or block the further reproduction or further the numbers, the increase in the viral numbers can be reduced. Fine. So as I was saying that, so this log is nothing but the proteins that you have seen before, the two proteins, and these are the keys. These two keys which are lying, we know that it is going to open this log. It's going to bind this protein, which is already approved by the FDA, right? Now what I have taken the right side is a bunch of keys, which are all keys of different, or the molecules of different or other than HCV virus. I have included HIV, virus, many viruses, drugs which has been available in the market. So I'm pulling everything together, the known key along with this one. I put everything together, and I trying to find the matching key from the other antivirals to the existing antiviral. Okay, I hope I have explained the concept in a simpler way. Fine. Okay. So first I collect the data set of the HCV and other antivirals from, okay, and I calculate or compute the molecular descriptors or the physiochemical properties, and then I do the clustering. The clustering is to cluster or group the molecules which are very similar to the known compounds. Okay, and then after that I take the other antiviral compound which is showing similar characteristics of the known HCV molecule, and I am studying the interaction analysis. So this is my antiviral data set, and all the dark colored molecule that has been represented here are the known HCV molecules, antiviral molecule, and the other molecules are from other antiviral drugs. So it is, I am putting the key along with the unknown keys. I mean, unknown keys, I want to find a similar drug which could be present in any of the antiviral drugs. Okay, so these are the list of molecular descriptors. So when I was saying about the flower, so color, right? Even the smell, or the length, the breadth, the petal, the number of petals, average number of petals. So all those stuffs, right? That comes in as a descriptor for the flower. So similarly, for every molecule, small molecule or a drug molecule, they'll be having a list of molecular descriptors. So as I was saying, around 4,000 odd descriptors are available, but we are considering around 2,000 descriptors. So these things have been calculated by using the Dragon software, which is also available free. So total 18 types and 2,168 descriptors have been taken. So once the descriptor has been calculated, along with the known HCV antivirals and with the other antivirals, so I'll be having a, so totally my time, I want to tell the dimension is, I'll be having 82 rows. For example, you think about the Excel sheet having 82 rows and 2,168 columns. I repeat once again, I have taken a pool of anti-HCV drugs along with the other antiviral drugs put together, and I have made 82 compounds, 82 molecules. So 82 rows in the Excel sheet and 2168 columns are nothing but the descriptors. So you think about the dimension of the data. So based on this dimension, see if it is one property, right, one descriptor or two descriptor, we can manually easily find out the, for example, molecular weight between two compounds or three compounds. So one molecular weight is around 500, first two compounds, second is 550, third one is 600. So we clearly know 500 is closer to 550, right, when we have one descriptor. So think about 2168 descriptors. So we have to use some softwares to cluster it based on every property or okay, of the 82 compounds will be compared with each other, which we cannot do manually. So we go to the some MATLAB or even hierarchy clustering tool from other statistical packages. So you can enter that and do a clustering. So clustering will be done based on the closeness between the molecules based on the descriptors, the calculated descriptors of 2168. So finally, once we have done the clustering, this cluster is a hierarchical cluster. So we see kind of a dendrogram like picture. If we do some other ordinary cluster, we'll be having data points clustered together. But this will give us a good clearer picture. So we have utilized this hierarchical cluster analysis. So dendrogram generated from clustering 82 antiviral inhibitors, and the vertical represent the dissimilarity between the clusters. Okay, so you can see the x-axis is the compound number. Okay, 82. You can find 82 compounds, including the HCV and other antivirals. Fine, and based on the similarities or the distance, the cluster has been arranged. Okay, so next is compounds obtained from clusters. Fine. So once the cluster has been done, so in the subcluster 2, okay, so this is a one major cluster, and the subcluster 1, 2, like that 3 it goes. So we have taken out the subcluster 2 alone, and we have identified what all the compounds that have been came close to each other in the cluster from other antivirus. Fine. So in the subcluster 2, we can see that HIV compounds are coming closer to HCV compound. For example, azinopravir is from HCV, which is closer to atazanavir. Okay. Another important point is, the valid point is, both of them are having the common target, protease inhibitor, because almost all viruses will be having protease as one of the one of the target in their genome. So based on the similarity, these two, HIV and HCV compounds, are coming close to each other after comparing 2168 compounds. And then if we go down, we can see that in subcluster 3, even cytomegalovirus, human cytomegalovirus, HCMV, and the ordinary cold virus. Okay, so all this virus and their targets, the viral targets are here. The compounds, the inhibitor that are the keys that we have taken, are showing some similarity between the HCV. You can see the HCV and HSV. Okay, so everything is present in the same cluster. So that's a good positive result, that we could able to find a similar compounds to that of the HCV. Okay, next is, so we have put the structural similarity here. In this case, in the subcluster 2, you can see that all the A's, right? If you look into the bottom of this picture, a star has been given. All the A's are HCV compound. So in the first dendrogram, this is a HCV compound. and this A is a HCV, this A. So totally 1, 2, 3, 4, 5, 6, 7, 8. So 8 HCV compounds are showing good similarity with other non-HCV antivirals. So it's a promising result that the key is, a little closer to the existing keys. Okay, so next, as you see, you can find indinavir. So indinavir is a HIV compound, which is very much closer to the HCV-A. And similarly, here also you can see AT130. So AT130 is from other virus, right? Okay. C means yeah, HBV. So we know that, right? HBV, HCV compound are getting closer in the subcluster 4. So next is a docking study for NS3-4A protease, where the compound name is we have taken is indinavir from HIV, and cyclovir from human cytomegalovirus, and atazanavir from HIV. So all these compounds we have, which are closely present in the cluster, showing good similarity in the physiochemical properties to that of the HCV, has been taken out, and they've been analyzed for the interaction studies. So the interaction studies, we have taken the protein with the PDYD ZZJO, and we have done the molecular docking study using this compound. So this could be acting as a reference compound, the HCV, the 2 HCV, and the 3 non-HCV antivirals from HIV, 2 HIV and 1 from cytomegalovirus. So as you can see, even it's just a computational representation, and it is showing good affinity compared to the HCV. The binding affinity of the HIV compound against the HCV protein target is much better than these 2 HCV compounds. So this is a finding. Okay. So, and this is the docking structure, the ribbon model of the protein along with the compound, along with the HIV compound, not from HCV, because HIV was found closer to the existing HCV molecule, right? And these are all the interacting residues presented in the 2D format. And the drug that we are showing is the indinavir. So what we are trying to say here is indinavir is closely, much better for treating the HCV. Could be, okay. So this is not a concrete result. This is just a hypothesis and prediction with all the computational tools. This has to be taken further to the experimental support. Just like making this keys a little bit of fine tuning the keys and making it more affinity to the protein target. And this is for the next protein, NS5B polymerase. So again, we can see that ritonavir and ribavirin, and then HSV. Okay, so all the other molecules are also having good affinity. Even RSV, it's a virus, other virus, and its compound is also better binding affinity with the HCV protein, the NS5B protein. Okay, so even the HBV, and these are all the amino acids that are interacting. So the same ribbon display and the molecule that are interacting with that, the lock and key, right? This protein is the lock, and this small molecule is the key, and these are all the amino acids which come into contact with this small molecules. Okay, so summary is, in this competition we utilized molecular descriptor based similarity search specific to HCV protein inhibitors from other existing antiviral inhibitors. So that I've been saying a lot of time. And next is clustering, hierarchical clustering of the selected 82 compounds based on the multidimensional features. As I was saying, the number of features is around 2,168 features have been identified from a few antiviral and retroantiviral compounds. The above compounds were filtered based on the binding affinity. So next filter, and therefore based on the cluster, it has been hypothesized that Indinavir and AT130 may also be considered in targeting the HCV protein with little polishing and refining, right? So because we have to it's not exactly the log, but much closer to the existing one or better than the existing one, as we have seen in the docking analysis. So as it can be transformed into potential compound for antiviral drug development, and we conclude this study will lead to the development of new therapeutic implication by improving the other antiviral and retroviral inhibitors against hepatitis C virus. And after that, I don't know. Wait, what happened to the publication? Okay, so this has been published in Life Science Journal, Elsevier Journal. Okay, so with this, I'll stop my talk, and I invite questions from you, audience. Thank you. Yeah, thank you.
Thank you, Dr. Febin. You began with a short introduction about hepatitis C virus, its structure, and the drugs used for its treatment. And you showed us a workflow for identifying the antiviral drugs, especially for hepatitis C, by using hierarchical cluster analysis and docking study for NS3-4A protease. Thank you, sir. It was a simple, concise, but it was useful to us. We have few questions, sir. Would you like to answer?
Yeah, sure, ma. Thank you, thank you.
Okay, so one participant has asked, what are the side effects of these antiviral drugs, especially when they are protease inhibitors?
The side effects are usually the ototoxicity and severe diarrhea. Okay, so these are all the major side effects that has been known till date.
Okay, okay. So the drugs which you are comparing, will it have the similar effect on the normal cells?
Normal cells, yes, of course, because only no, all the side effects or the adverse effects is because it is interacting with the other cells also. Even the anti-cancer, all the side effects. Any drug, if you take, apart from acting on its own target, it will again act on other cells also, and it will impart this side effect in that way.
Okay, okay.
Yeah.
There's one more question, whether these compounds are drawn in ChemSketch software or from NCBI Chem.
This is not drawn. It has been directly downloaded from the PubChem as a
Okay.
SDF format, SDF format or PDF or PDB format. Many formats you can download directly,
Okay.
and you have to optimize the structure to get a better 3D view. Energy minimize it. All pre-treatments are available. You have to apply molecular mechanics to these drugs. Yes.
Okay, okay, sir. One more question. In
Yeah.
slide number 15 that you had
Uh,
shown, why are there no clusters in cluster 1 like in cluster 2? Why there are no subclusters in cluster 1 like in cluster 2? What is the basic difference? Why is it showing two different clusters?
Subcluster. It's not distinctly visible, or no, what is the question?
Why are there no subclusters in cluster 1 like in cluster 2?
Okay.
That's it.
So cluster one. as you can see, the cluster 1 compounds are all very close to each other, and they are very distinct, because one thing we can infer from that is all the compounds are protease inhibitor. They all target the same protease from different virus. So that could be the reason for that. And if you go further, after that, you can see different antivirus, the viral compounds coming in the cluster. That could be the reason.
What are the side effects of Insinar?
No,
That's a question from the
I can
participant.
only say the general side effect, because we have not looked into that. No, every compound specific. Maybe in the future we'll be looking into the side effect specific analysis like this. Okay, thank you for the question.
Okay. Is there any possibility for making antiviral drug using docking method for corona?
Antiviral drug. So we all know that even the Glenmark, a company that is giving a drug, Flavipravir. Flavipravir has been used for some other antivirus, and they are still giving this one. So since all the antivirus are having no common drug targets inside, for example, protease, reverse transcriptase, so it may work moderate to mildly, but not having full swing or full potential. It cannot work. Using docking alone, as I said in the beginning, bioinformatics is a complementary field where it can complement or help or aid in the experimental or reduce the time and money that has been, for example, to develop a drug. It takes around 15 years, 10 to 15 years, and $1 billion dollars, and even after coming out it may fail in the market. So these failures can be avoided if we go through the bioinformatics approach. For example, without knowing that it is a toxic compound, when people are working in the experimental laboratory, and they spend around 5 to 6 years, and later they come to know it is having toxicity. So instead, there is a toxicity database which is already available. So you take the compound first, whether it may be isolated from a herb, or you synthesized it or whatever. So take that and compare it with the toxicity database. And if it is saying toxicity is there, you can avoid using that, or take it to the further experimental approach. So docking alone is not the only. It can reduce the time and money, as I was saying.
Okay, okay. One question. How do you
Yeah.
detect? I don't understand the question. Who's Saravana Kumar Setu? Can you please question again or unmute yourself? How do you detect of drug to involve to level incubation? Do you find any meaning in that, sir? How do you detect of drug
I'm
to
not
involve?
getting it. How
Yeah,
to?
even I'm not getting that question, so we'll skip that question. Okay. Which docking software did you use for the study?
Autodoc. Autodoc.
Okay.
All are open source only.
Okay, 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 sir.
As
And
I
one,
was saying, no, see, since in this Corona period, you
uh,
all will be in the home, and you all have a laptop and the internet connection. You can start your bioinformatics research. There is
okay,
no bar.
okay,
Nobody can hold. Everything is so open source. Of course, the proprietary software is also available, like a Discovery Studio, Schrodinger. But all this, there everything has been packed in a single package. But here, if you go open source, everything will be found in a bit by bit, block by block. Okay, so you can use it and even publish papers. Yes.
okay, okay, sir. The protein 2ZJO is refined or modified for this docking study.
No, it has been directly downloaded from the PDB database,
Okay,
and the energy minimization and the pre-treatment that is available in the Autodoc only has been followed. We
okay,
have not followed anything
okay.
other than that.
Okay. If there is any new drug that is being manufactured, will there be any cross immunity? For example, if there is a drug between for HCV and COVID-19, Will there be any cross immune reaction that is happening?
Can you repeat, please?
Will there be cross immunity reaction happening in the body between
Mm-hmm.
HCV drug and a COVID-19 drug, if at all it is identified?
Uh, um, yeah, cross in activity is class. So
Okay.
cross activity and the increasing the immuno activity
Yeah.
is all there. Okay,
Okay.
it's called drug-drug interaction.
Okay.
When two drugs, even if we take calcium, I mean aluminum, for example, a simple example could be if you take milk
Mm.
along with aluminum hydroxide. So there is a formation of calcium hydroxide. Aluminum hydroxide you are taking it for, or magnesium hydroxide you are taking it for ulcer or gas trouble. And along with that, if you consume milk, so there will be cross reaction. So obviously, so there could be possibilities are there.
Okay.
Yes.
One last question, sir.
Hmm.
Why is the
Okay.
preparation of COVID-19 vaccine taking a lot more time? What would be the general procedure for preparation of vaccine?
See, the very good question. There is a time limit, right? For example, as I was saying, for a drug, it takes 15 years. There are different phases of
Okay.
drug development. Screening, phase one, phase two, animal testing, human testing, then approval, then you have, and whether the sufficient number of immunoglobulins are
Mm.
produced for the COVID.
Okay.
All this are no different angle. It has to be worked out, and that's why it is taking many more time.
Okay.
I can also tell you that there is a immunoinformatics. So if no, after this session or after today, you can go to the immunoinformatics, Google the immunoinformatics, and you can find there are many. I mean, actually, before it was the vaccine are all like half-killed vaccine, right? For example, BCG, tetanus, all are half-killed virulent vaccine. But now it has been completely different. It's all now subunit vaccine. So from the complete protein, instead of the organism itself, species itself, they take the individual protein. For example, this protease protein, and from that they will be taking a peptide out of it, which is highly antigenic, which is the one which is going to bind to the MHC protein, major histocompatibility protein, and then there it binds, and the downstream process, right? How it invokes the immunoglobulins for the certain peptide. So it's all now peptide based vaccines. So mark these words and you can search it, and you can also do it like I have used different tools. So peptide based or subunit vaccine or peptide based vaccine or immunoinformatics, if you use these keywords, you can try to find a solution. Anybody
Okay.
can find, right? Yeah,
Okay.
good question. Yes.
Okay. Do hormonal problems lead to HCV disease?
No. Hormonal problem, no.
No.
As I said, no. This is completely person to person. So in southern region, this is not. No, we are all clean and neat, and so the plates. We are not sharing any. Sharing needles. Only those people will get, not hormonal and all.
Okay. Okay. Uh, uh, in PubChem database, is SMILE format good for docking studies, sir?
Smile format has to be converted into 3D format.
Okay.
Smile is a 1D, one
Okay.
dimensional, two dimensional, three dimensional. Yes.
Okay. Okay, sir. Okay, thank you, Dr. Febin,
Yeah.
for
Thank you very
being
much.
making yourself available for this session. Thank you. We'll meet soon, sir. Thank you.
Yeah. Thank you. Thank you all the students who patiently listen, and thank the principal of St. Xavier, the supporting staffs, and of course Mabel. Thank you very much. Thank you for the time. Thank you.
Thank you, sir.
Bye.
next.
A very good morning to the participants. We are waiting for the next resource person. We'll resume the session in 5 to 10 minutes. We'll get back to you. Please be on the line. Good morning and welcome back to the second session. Our resource person is here. I would like to introduce our resource person to you all. Thank you for your patient waiting. Dr. L. Ranjit is a resource person for the second session. He did his bachelor's in fisheries science in Fisheries College and Research Institute, Tuticorin, a master's in fisheries science and PhD in the Central Institute of Fisheries Education, Mumbai. He has nearly 9 years of experience in fisheries resource management, taxonomy of fin fishes and shellfishes, cnidarians larval biology and taxonomy, ecosystem-based biodiversity and conservation, scientific underwater dive, biodiversity loss and economic valuation, conservation mariculture, remote sensing, and climate change. He is a trained scuba diver and he is a recipient of Professor N.R. Menon Memorial Award. He has cleared ICAR Net Award, Institutional Fellowship by CIFE, Institutional Fellowship by ICAR for pursuing his master's, and Dr. Hiralal Chowdhury Gold Medal in his master's. Altec Young Scientist Award also. He is currently involved in in-house projects. He has completed 6 and 4 are ongoing, and external funded projects 4 completed and 2 ongoing of CMFRI. He is a member of 16 national committees, published 1 book chapter, 38 research papers, 25 technical articles, 2 reports, 4 technical bulletins, 64 abstracts in international and national conferences. His salient research achievements include depositing more than 25 specimens in the designated marine repository of CMFRI and GenBank submission of more than 65 specimens in NCBI. He has done underwater studies in assessment of coral reefs and associated fauna from Lakshadweep Islands, Karwar Islands, Park Bay and Gulf of Mannar Islands and the Watch Bank, India. And he has reported mass spawning of corals from Lakshadweep Islands. He has documented different jellyfish species diversity and peak scyphozoan swarming periods along the southern part and the Watch Bank. Dr. L. Ranjit started his scientific career at NARM Hyderabad and is at present a scientist in the Fisheries Resource Management, Marine Biodiversity Division, Tuticorin Regional Station, ICAR, Central Marine Fisheries Research Institute, Toothukudi. And currently, he is carrying out research on the scyphozoan and cubozoan bloom dynamics, taxonomy of sponges and sea anemones, marine viruses and bacteriophage diversity, delineating biologically sensitive areas and assessing resilience potential of coral reefs and other sensitive ecosystems. And we are feeling honored and privileged to have such a wonderful prominent person among us today. We welcome you, Dr. Ranjit. You can start your presentation and the session is all yours.
Okay, thank you, ma'am, for the nice introduction. Hope I am audible now, ma'am. I am audible?
Yes, sir, you're audible, sir. Clear.
And my screens are visible now?
You have started sharing. Yeah, now it's visible.
Visible. Okay, it's fine. Nah?
Please put it in full screen. Okay, thank you, sir.
So, one minute. Yeah, very good morning to everybody. First of all, thank you, this organizing committee, for inviting us a resource person in the zoological seminar.
Excuse me,
and,
sir.
As
Ranjit,
it's audible?
Sir? Yeah,
yes,
it's
yes.
audible. Can you turn on your video, sir?
It is. Not visible?
The video icon is enabled, but we cannot see your face. Okay, no problem. We'll continue, sir. We'll see.
Okay. We will continue as slides are visible.
Yes,
thank you.
Okay. Thank you, sir.
Thank you very much. And my lecture contains, I think this zoology seminar will be a wide audience, starting from students and to the eminent scientists. So my understanding, because this zoology is a subject which is base for everything, like from botany, like biology, zoology, and botany is major streams. Then later case, during advancements of science, then it deviated into different forms like biotech and even fisheries, everything. So we will start with certain basics, because we should also know some basic, because now things are changing. People are more thinking about the latest technology, forgetting everything about the basics of science. So then in later case, after that we'll go to the Nigerians, because I have some insight experience on some of the Nigerian resources. And finally, as Madam told, we have the sting jellyfish things, and a small meta-analysis through these viewers and Web of Science knowledge, and what is the current status of this stings and innovation. So starting with that, so we have starting with the hierarchy of biology classification. This called taxonomy. Who this Linnaeus is the person who initially starts with this biological classification, starting from the life to the species. As I already told, we have a broader kingdoms like plant and animals. So that is that botany and the zoology. So even Linnaeus itself knows these are the major key source, key subjects which has to be focused. So as I told, it has been diverted to different fields in different areas. So then I was thinking, as Linnaeus told that it is life. Then we try to find out some cartoons. What is life? Is it difference? It's difference between the life stages between the birth and death, and then one more interesting image I had to find. What is the meaning of life? Now all go to the digital worlds, including we organizing many kind of webinars and many meetings through digital mode. So even a basic thing, if we ask, somebody will respond, I don't have a computer or internet to respond. Such status it went. So this is a small clips where the life is nothing but the study of biology. Study of life is a biology. Life. So there are different components by some Chris, where life. Another thing is alive. Where the animals or any plants which having growth for development, for reproduction, response to the environment, and cellular and structural composition, and those animals which perform metabolism and homeostasis. So these, whatever animals which perform, these are the activities, these are considered as living things. So we studied in the early stages like cellular level, where we have only three domains previously, three domains were archaea, bacteria, and eukarya, and the non-cellular level, the latest things are prions, virus. Moreover, this kingdom of classification, we know that initially the Carolines we have two kingdoms, later it is merged to five kingdoms, now it is six kingdoms. So latest, as per the latest reference I got, Rodrigo, he proposed the seven kingdoms. Though many kingdoms of classification exist, but this is the latest one. What I found, where I found it is that's prokaryotes, where bacteria and archaeas are involved. These all are unicellular, and the eukaryotes mainly the multicellular. Some are having some exceptions, starting from protozoans, promestis, brown algae, plantae, fungi, animalia. So this part, our animalia is it's our of our subject zoology of concerns. So most of the things when we see the seven kingdom classification, the animalia major forms 36 kingdoms representing diverse forms. That what we are studying is that zoology. So when we see the characterization of different animalia classification, there is some kind of level of organization for in an animal body. Like it may be organ level to the protostomic level. So, for example, if you see cellular level, most of the sponges comes in cellular level, tissue level, coelentrates, and organ level, platyhelminths. Just a gist of that, because when we tell some subjects, sometimes it may confuse. Many of them are knowing the things. So another thing, when we comes with classification, whether this animal is diploplastic or diploplastic, is nothing but the number of layers, germ layers found, like maybe ectoderm, endoderms, and maybe mesoderms. And some are classified based upon the symmetry, whether asymmetrical or radially symmetrical or bilateral symmetrical. Then some we have a coelomic structures, where they are acoelomite or coelomites and pseudocoelomites. So these are the basic forms which forms a classification. This also includes the segmentation, where it is platyhelminths having some segmentation, even crustacean have some segmentations. Most of the arthropods also having some segmentations. Then apart from that, notochords, where whether they have animals have some notochord system, there's chordates, those animals which don't have any notochords, non-chordates like poriferans and some echinoderms. So coming to our major subject, like phylum Nidaria, many people might be knowing this from the reports through the fossil studies. It originates late backs in Adcarian Carinae period. Now, recent molecular tools suggest it is maybe of 1 billion years ago. That means 110,000 millions. So, what is the peculiarity in this phylum Nidarians? Because when we observe or in the field, these are really very beautiful animals. Because I'm being a paddy diver, when we see the animal in 3D world, it is really beautiful. So, where there is some progress in where whatever some beauty is, and some we should be very little precaution. My father used to tell. So beautiful, it means something, it may cause deadly animals. So cnidarians, when we see as a whole, some of the cnidarians which cause deadly, like the Portuguese man of war, that's Physalia, and some are colorful and varied. Many people might be done underwater surveys, where if you see it in underwater, many corals, that is stony corals, anemones, and gorgon, it looks very beautiful, very beautiful. And some of the animals are very small and highly abundant. We can't see much in our naked eye, some are animals, and some are delicate, like if you see the jellyfish, it major part will be content of water, so delicate, and it moves in a massive blooms. So as a whole, cnidarians in terms of ecology, we can tell that it are considered as producers, because some of the corals which having a symbiotic algal forms, because they have symbiotic relationship with the algae which forms most of the reefs, and some of them are massive, like they form a production mass blooms huge density in certain periods. And some of the jellyfish even occupies a tropic chains in high level, because they are exclusively carnivore species. So these are the some important features which decides the cnidarians. Then coming to our these characteristics of phylum cnidarians, because when we see that cnidarians is a specialized stinging cells called cnidocytes or nidae, which is where the term phylum Cnidaria creates. And apart from that, it has exclusively radial symmetry and a diploplastic germ layers with mesogel, and then they have a blind get. Some interesting that they don't have any respiratory circulating respiratory systems, and have a complex life cycle, because they have both the polyps and medusiforms, and polyps and planula in the development stage, and many colonical forms have exist polymorphism. As all knows, the radial symmetry exist. Where, for example, first one is the polypioid form, second is the medusiform forms. When we cut every side, it divides into eucal forms. So this indicates the radial symmetry. And apart from, we have germ layers, two germ layers. I told it has don't have any prominent mesoderms, but we have ectoderms and we have endoderms. Okay, and then as I told, the mesogel, it is a acellular portion where uh, it has a jelly-like. This mesothelial gel is nothing but a jelly-like substance which consists mostly mucopolysaccharides and some collagen-like fibrous proteins. In most of the, for example, if you see the jellyfishes, mesogel, it's mostly found in. And when we see this role, it can mostly elastic properties which helps in contraction and the movement of that animals. And apart from the mesogel has having some wandering amebocyte which having some debris and bacteria for phagocytosis. As I told, they don't have any excretory circulatory respiratory system. What happens, how it sees, or how it goes, how it moves in most of the forms. Okay, So generally they have a cavity called the gastrovascular cavity, and as a whole, if you have seen, it also has some blind gut sac because it opens in one side only. It closed in other side and opens in one side. Both the mouth and anal part is exists in the same portion. So it generally terms as a incomplete digestive tract where food will be taken, get digested, uh, and get moved out. So this blind gut sac or this gastrovascular cavity plays a major role in the digestion, circulations, and distribution of nutrients and oxygen transfers, etc. Apart from some of the animals having this gastrovascular cavity has succinylate in it. Then, then when we have this cnidarians, mostly it has a sticking cells. Okay, fine, because there are many types of cnidaria exist, but most prominent in all the cnidarians is the cnidocytes, where it has stinging cells. Will have a trigger-like mechanism which discharge some kind of threads and inject the venom into the animals or any personals. So apart from that, we have a complex life cycle. As I told, some of the jellyfish has a metagenetic life cycle, which have polyp phase, medusae phase, and some of the polyps stroblates and use effray. And during sexual reproduction, they form gametes, and the glandula is the larvae produced, then get settled, and such a very complex cycle in the jellyfish. Because what here I also highlight some of the cycle because I also worked on this larval biology of some of the few jellyfishes and established some larval cycle of one species of Cassiopeia andromeda, and I'm working on another species for Mastigasis papua. So first thing is here we found the polyp via stroblations, which I found it. This is already communicated and it is under process. Many young effray it found stroblated release effray. Then we moved to young effray, advanced effray, meta effray, young medusae. This is what observed in our laboratory. Finally, the young medusae. So it almost having a typical stage of 45 to 60 days forming a young medusae. But most of the ephemeral stages exists only 3 to 4 days in our jellyfish effray, what I observed. This is a Cassiopeia andromeda, which is observed in our laboratory. Another thing coming to our diversity angle, because everybody is thinking how many species exist, how many numbers, how many are toxics. Because generally cnidarians mostly divided into two subphylums. One subphylum, under one subphylum, that belongs to Anthozoa. It is more diverse species where these corals and anemones exist. Another which hydrozoans, mostly this hydroids, hyphenophores, fire coral exist. Approximately 3,500 species exist. And the class hydrozoan, cyclozoans, almost these are the two jellyfish. Nearly 200 species exist. And cubozoans, most of the box jellyfish and sea webs exist. Nearly 50 species exist. And another class comes the staurozoans, where the stalk jellyfish, nearly 50 species exist. These two class, the class Mixozoa and the class Polypoidozoa, because worms, most of the literatures we found this class Anthozoan, Hydrozoan, Cyclozoan, Cubozoan, latest is Staurozoan. But recently this Mixozoans is added in the worms registry. So mostly these Mixozoans and Polypoidozoides are parasites, parasitic in nature. This Polypoidozoides recently this Chang has included, but in worms register, that World Registry of Marine Species, it not comes in accepted form of class. So In total, almost six classes exist, where this class one is class Polypterozoa is it is under the revisions for making in the phylum Cnidaria. Okay, then we will see one by one what are the major key key characters of these hydrozoans. As I told, hydrozoans are almost 3,500 species, where it is mostly have a polyp and medusae stage in the life cycle. Although where the polyp stage is dominant, the polyp, cavity is simple not divided by mesenterics. Generally, the mesenterics decide the symmetry, and in corals and all they form septum, which helps in the identification. And the oral coat has some elevated parts, hypostome, and in most of the gonads is ectodermal. In Medici stage, where we have balancing scatosets and photosynthetic organs like ocell. Then these hydrozoans are mostly solitary and colonical form, and they have highly polymorphic. For example, if you see Portuguese, they have nematophores for floating, etc. And generally, the denosate is released only through via operculum. We don't have any cilia. And here one we found that Cladonema radiatum, which is found in our copypod culture tank, because this Cladonema, it is in the sea taxa. We have scientific sea taxa. They found in mostly the copypod culture tanks. Next, some major classes exist in hydrozoans. This subclass is divided based upon the origin of where it is located, the location of the datosets. And then we'll come to the class cubozoans. These are the box jellyfish and the sea crabs. Because this box jellyfish is topmost venomous species, where it has medusae and polyps, but medusae stage are dominant. And the polyps don't have any internal features because there are no kind of mesenteries, everything. The peculiar features, it has some velarium in the margins, velarium in the margins, and the mostly we have gonads in the ectodermal region. It has specialized sense organ like Rophalia and scatosets. Generally, the mushy, fleshy plaque are the pedalia, which helps for this holding of most of the tentacles. And they have major groups like order Charybdis and Corydotia, where based upon the tentacles only it was differentiated, because Charybdis is having single tentacles, and whereas other order has more tentacles in the pedalia. And we have there is no gastric sepals in the Caridia, vice versa. This cubozoan really extremely painful and having many fatalities to humans. Even our place, our location, we also reported some fatalities in due to cubozoan sting in nearby region, our Tuticorin region even. And they are because mostly preys on smallest vertebrates. Even because every in food chain, it also has a predator. Some of the butterfly fish, butterfish, batfish, some crabs, turtles eats on cubozoans. Next, it comes to the another groups are the scyphozoans. Scyphozoans are nothing but the true jellies. Here also mostly the medusae stage is dominant, and it has a polyp stage. The polyp stage undergoes some stabilization-like process. So stabilization where it is release of ephirase. Maybe some of the polyp may be stabilization may be monodiscus, some are polydiscus, where it releases more number of ephirase from a single polyps. The peculiar features, it has some clefts where it is reflexes, and it has projections, laphets. When compared to the cubozoan, this bell margins don't have any velum in the scyphozoans. Okay. So finally, they have some guts which has complex radial canals, right? Then similarly, this unlike similar to the cubozoan, it has endodermis in the gonads. and it has Localia and Ocelli and Scatosids. These are some structures of scyphozoans. We also reported many scyphozoans along these regions. Mostly the scyphozoans are two major subclass, the cornate medusae, mostly crown forms in the tentacles, and discomedusae, and under discomedusae we have rhizostomine medusae and stemostomy. Because based upon the number of oral arms only it was classified, because in many of the rhizostomine members, even scyphozoans, it is useful for in human consumptions, because in many of the Southeast Asian countries, like some Rofalima, has been used in the consumption of jellyfish. Even we in India also, in India, some of the states like Gujarat and Visakhapatnam, we have a commercial fishery of few jellyfish species like Ramberlina arseni, and where the tentacles and oral arms has been processed for various purpose. Similarly, in Scatostylus, some species in Gujarat has been, we are also reported in our project for fishery of this. Mostly it exported via our mostly Tamil Nadu, Chennai, and some are in Pipava into Gujarat, exported to mostly in the Southeast Asian countries. And here in some part of Kanyakumari also have some fishery. This is about scyphozoans. Next we comes for the anthozoans, because these are the very beautiful, I mean, the flowers animals. Here only difference with other animals, other classes. Here only the polyp stage exists. Okay. Then moreover, the gastrovascular cavities is divided into mesenteries, which decides the uh, body wall. Radiated inward is the body wall, decides the symmetry of the animals. So based upon the symmetry, either maybe hexamerous and octomerous, like octocorelia or hexacorelia. Then here both the operculum and cilium is absent in nidocytes, unlike other other classes of cnidarians. Here they don't have any operculum cilium in the nidocytes. So some of the broader groups of Anthozoa, these are the very beautiful numbers. Many of the scientists are working in different regions, mainly the corals, exclusively beautiful forms, stony corals, scleratinans, and the sea anemones, wherein many have aquarian potential, stings to the half of the humans. The amphibians, some of the anthozoan, most of the anthozoan members like corals, some fire corals, all are in list of Wildlife Protection Act. And we have coralliomorph, which we have coralliomorphs, and zoanthids. It almost looks similar to corals, but it don't have any hard structures. And then we have octocorelia, which having 8 mesenteries. Under that we have soft corals and gorgonids, and we have this Heliopora. They are blue corals. The order Pentacula, the sea pens are sea feathers. So these are the complex groups exist in the anthozoans. Right. Then right. Then is class Starosova. Starosova is toxic jellyfish. Is generally we have a Staromedicine groups. They don't have any alternating medicae, but it has some polypoid voice. Generally it looks in upside pattern. Moreover, the cilia does not possess any ciliation is present in the planula larvae. Only few numbers has been reported in the Starozoans. Then other two groups, as I told, this Mixozoans, these are mostly nidonospores, Mixosporidians. They found mostly in freshwater and marine, because they want some host to life. Because in general, most of the cnidarians are marine, and few hydrozoans are having freshwater, and they have a brackish water influence also. But this obligate paras, the nidorian force exists both in freshwater and marine force. They have some kind of post life cycle involving in fish, and some are found in the annelid worms and bryozoans. So they have some symmetry, spores formation, later it transformed into nidarian nidospores, which enters into the animals. So next one is the class polyphysoid, because these are also similar to the parasitic nature, but still taxonomic confusion exists. But it has been reported by some reported in some of the sturgeons and paddlefishes. So unlike it has some animals which lives in metazoid forms, then host eggs in be mostly these are freshwater, because sturgeons and paddlefish are mostly freshwater origin. So they have a peculiar features of cnidarians. Maybe in future studies will be conducted, it will be maybe placed in a prominent class of under the phylum Nidaria. So these are some of the beautiful representations, some of the cnidarians, starting from jellyfishes, ends with hexacorallia, and this is another reports showing different cnidarian representatives. So unless other groups, these cnidarians are more beautiful, and the second part, as I told, it's somewhat stinging, right? Because they have peculiar nidocytes or nematocytes, which serves as a defense or even harms or even kills the prey or predators. Okay, because generally these nidocytes having it won't harm every time, because sometimes some animals approaches, or some swimmers or divers approaches certain animals by knowingly or unknowingly. So it has some, as I told, the operculum, it has some operculum. uh, this having a nidocyte is having some capsule like wall, which discharges due to some kind of trigger mechanism. Then the operculum exists, where the forcing tip comes out. Then discharge exists. It having some kind of spines which pierce through the skin or any other animal epidermis. Finally, it releases the venom. So this is a pictorial representation of a stinging mechanism in humans, because as when we do some scuba diving, or when we do some collections in nearshore or open waters, we should be very careful with these cnidarians, because it can, some of the cnidarians can inject the venom in a very fraction of 30 milliseconds, because such a fast thing, it can drill our pores, or drill our pores our epidermis and inject the venom. This is a simple stinging mechanism which is adopted from Lawton et al. And when come to the cnidarians cost to the humans, because almost 11,300, in if you are including mixozoans, it's almost 13,500 animals exist, of which 22% are dangerous to humans. So the top nearly almost 200 species, the top species are the Portuguese manafor, the Physalia. Even it exists in our Indian waters, and some mallow. These all are found in mostly Brazil and other countries. And the box jellyfish also found in our Indian waters, which cause harm to the humans. Then if it comes next, other groups like Phycoris, millipose also harms to humans. Then sea anemones, like hell's fire sea anemones, night sea anemones. These are the some of the animals which causes injuries to humans. And apart from that, some venomous nature, because most of them, as more 200 species exist. Some of the representative species from author Montgomery, I have shown for your reference, for list of hydrozoans, mostly Physalia, and we have Scyphozoans, mostly the rhizostomian members. Rhizostomy and stemostomy members are very common in our Indian waters. We have Chrysora, Sine, Chrysora, Pelagica, then Rhizostomy, or even Cassiope andromeda. this species also cause sting, because these Cassiopeia andromoda, it's like an outside jellyfish. They release some kind of caseosome, which gives sting when the caseosome release in water. Papers also that on report of caseosome in nature, where it causes a sting to the region where animal may not exist, because that kind of caseosome has a report where cause harm to the human also. Next is the cubosovans. As I already told, cubosovans are very toxic. Some list of species that's mentioned, and some andosovans, and like sea anemones also listed. So come to this compounds, because generally when we considered any toxin or venom released in compounds, there's some kind of pharmaceutical value. So based this report I have taken from Sanjanam sir book, where we have many terpenoids, because most of the pharmaceutical compounds found in the marine ingredients as a whole are mostly terpenoids. Because I have not much worked on this region, but for insight to the audience, because many of the students are working on this compound activities, everything. So these are the major compounds of marine ingredients, like then followed by steroids, eicosanoids, and up to lipids. Then bioactive compounds, because marine, whenever some compounds are there, they have some bioactive potential. So mostly anti-tumor is 41% of marine ingredients have this anti-tumor potential. Some are having antimicrobial, anti-inflammatory, and anti-ulcer, and others. So these are a nutshell about this. And some of the groups which having therapeutic value. So there are different groups of species which having therapeutic activities. Some are can take and work on this species for finding out the therapeutic activities. And then will come to our sting, because whenever sting exists, whenever some kind of this nidarians, we have to document these stings, because in India, if you are concerned, we are not much documenting or focusing on the sting, because in unlike other countries where jellyfish forms major stings globally, there are reports 150 millions of people has been reported with jellyfish stings, either as recreational or through like bathers, swimmers, or some are through professional divers or fishing. So these stings as documented much in other countries, but we are a little lag. So we have to find out some ways to document or make some platform to document most of the stings, or in general as a marine stings, and in more specific as jellyfish stings. These are the some of the countries and cases reports on global records on jellyfish stings. And similarly, the marine nidarians were different species contributing their fatalities, because as I told, here in India also some species got fatalities to humans. So the major groups is the box jellyfish. When compared to other box jellyfish forms major groups causing fatalities in different parts of country. Finally, other like Physalia, Stomoplophus, and many species which causes the fatality. These are the dominant species causes fatalities. So another part, I was trying to find out how this Nigerian research is going on, because I used this Web of Science of ICR uh, uh, subscription, where what was the trend in going research in recent trends, recent decades? Because almost when I searched the term using Nigerians or Nidaria, I found that almost some 5,592 publication from the Web of Science, Web of Knowledge, Clarivatic Analysis. So when we see the trends of during from 1998 to 98, almost the number of publication, it has some kind of increasing trends, because 2020 is less because it has taken from half up to June only. So it is less, but the trend in going the research of Nigerians is go on increasing. So that's the progress exists in our global Nigerian research. So in even in India also we are working on mostly on different parts like orals. Still we are there, but when compared to the global scenario as a whole, Nigerians research we are in a little lag. So then I found what are the publications which found having more citation in the Nigerians. So these are the other barcoding of animals and classification of ecological data, because this will take wherever the keywords Nigerians exist. So some of the citations of citation, you see almost published 4,198 citations found it, and different top 10 papers and relevant authors and journals, years and citation has been represented when we view from the Web of Science knowledge. So these are the things has been cited in a bigger way. And then I am seeing what are the subjects people are working globally. So when we see in the Web of Science categories, we found mostly the marine and freshwater biology. So equally it is found in freshwater also. The research is going, and mostly the head is zoology, ecology, and even in fisheries almost 128 like jellyfish fishery. So these are the broader research areas where the Nigerian research is going on. So starting from the ecology and ending to the neuroscience. So various forms the Nigerians, one or other forms has been researched. Then in that documents, I was finding out whether we are finding this, even animation and stinge. I use the term even animation and the term sting. So who all are all working in this much area? What are the key terms used? For example, when we use this animation, mostly environmental studies has done much on the jellyfish and many other forms it has been studied. Similarly, when we, in the term of sting as a whole, Nigerian and venom aspect, it has been studied. In terms of publications, where citations of this Kurzal, and there are many authors, the bubble increase, the size of the bubble indicates the number of increase in citation of the papers. So similarly, evaluation, there are a humpty number of authors that Maritina, that publication has more citations, and the Girish Veena, who is an Australian scientist who is working much on the evaluation aspects. Similarly, these are the list of scientists is working on sting and even animation aspects. Then I was finding whether we when we see the global trend, we are not in the figure, whether our citation is not there or we are not having proper funds like that. I was thinking, because when we see that the number of publication is an output, the National Science Foundation and many of the institutes, none of our Indian funding agencies, which in terms of publication, because that is different. I'm not blaming. We have also fundings from many environment, forestry, CSR, Institute, DBT, DST funds. When we come the global level, in the terms of output, most of are not in the figure. When we the top 15 we are considering, and then we are seeing that this is not for criticizing, because the trend how the global research and exists between our globally and the status of our country. Then when we see the organization involved in the most of the Nidarian research. For example, University D is so lower because this having some my two friends, which is more Andre, who is working on Nidarians, because this institute having the maximum outputs, right? So, for example, 246 papers on Nidarians, the word Nidarians between the 1998 to 2020. So, some of the Spanish countries, then James Cook universities, and these are the major country universities working on this Nidarians. Then because the ultimate aim is our publication. So whatever research we do, we want to publish, so publish or perish. So such way I was trying to find what will, because in general we all will do a good research, very sincere research, but the selection of journals for aptitudes towards which journal we have to publish, that everything is required for getting some good impact in international ratings. So when I see the publication of Nigerians, the Zootaxa journal, because Zootaxa recently many are naming it has been removed from JCR. So there are because Humpty Nanda scientists has worked on this taxa, Zootaxa. But I want to hint something, because Zootaxa, when we come the basic taxonomist, we always prefers some taxonomic journal like Zootaxa. So even it is somewhat heartbreaking where this Zootaxa is not in recently in JCR list. So others, it is other part. So we are finding out the different journals where exist for the Nidarian research like Zootaxa, PLOS, then the Journal of Marine Biology Association, Zookeys, these are the top 25 one. Then what top I mentioned is one is the eve animation and the term sting. The eve animation term used has been mostly in the PLOS one, whereas the word sting used in mostly the journal Toxicana. So whatever I mentioned, the top is mostly the eve animation. First one is eve animation, and there is the sting term. So this is this bottom one is mostly on the journals as a whole, Nidarians. So these are the some of the key journals. We have Coral Reef Journal, Biodiversity, which can contribute our research items. Okay, fine. But the major chunk is by the Zootaxa and PLOS one. Okay, and next when we see the meta-analysis for country wise, right? So because I want to find our country, some way we have to brought it to our country, whether we have yes, we have also a reasonable contribution for in Nidarian research, because I because the projection I made is using a platform where the term Nidarian, because when we use different terms we have get different publications. So as a whole, if you see, the US is the leading country which is working on the Nidarians. So almost India is almost in 28th position. So followed by Germany, Italy, Brazil. Yeah, Humpty number of works in Brazil, Australia. But when we see in terms of eve animation and sting, mostly the eve animation aspects, both are similar term, but the usage will be different. Mostly work on the Australian, as I told, that the Grishman was working most of the things, and the sting mostly US, and the other countries it is represented. Okay, so these are the countries representing the Nidarians research. So our experience, because we have also worked on some of the jellyfish diversity and some first aid sting, and some kind of package of practice for initiating an app for jellyfish, starting with the jellyfish, more specifically on the cubozoan and scyphosovans. Okay, because in if you see the jellyfish, we have a metrics developed by some European countries, and we have jellyfish pro apps for identification, everything, where they feed the jellyfish sting also, where and all the sting exists, where the bloom exists. So here the toxic jellyfish I will be highlighting, or the major Physalia, major groups representing the sting. Okay. So what happens when we diverse symptoms? Because mostly dermatitis, this I took in a reference book, where it has some skin problem, because whenever we go, because all are won't much harmful, as I told you earlier. Initially, it have some skin problem, dermatitis like this, and some of the stings which can immediately like death. So these are the major problems like skin problems, abdominal like irritation, rashes, red swollen markings. Then some will create the abdominal problem like nausea, vomiting, diarrhea. Some will have this breathing problems like breathing difficulty, respiratory like pain in chest, running nose. Other associated major problems are like sweating, pain in muscle, raise in body temperature. These are the major symptom when we found encoding in kind of jellyfish sting. Okay. So generally, there are we have some they have package of practice. This is not our result. This is generally the first aid practices practice in other countries. So let it have to be get out of water. Some of the jellyfish we can apply vinegar. Some can apply hot waters, and some we can remove tentacles with forceps, because you should not scrap if you have any sting. You should not grab, because the nematodes just still discharge will be more, and it get injected little fast even after the animal is not there. Okay. So generally, whenever these are all the slides from foreign countries where they have emergency system where some association exists for some kind of advice for the jellyfish sting. So the better way is we'll have some kind of precaution material, because jellyfish and all will have some seasons for season for blooming season. So at that time we should not go for swimming or bathing everything. Whenever in seashore, because it will be stranded over there, you should not touch in barefoot. Always when we go with underwater, we should have wear a full wetsuit. These are some of the tips exist, and this is where our observation in the jellyfish sting, because here also we are, while working on our jellyfish diversity and this jellyfish sting, what we observed is some because box jellyfish generally occurs in the invertebrate region like shrimp resource area. So some of these shrimp collectors from Inigon, our Tootukudy personnel, they went for catching shrimps by hand picking. So what the victim told us, it is Physalia, because we found that later it is not Physalian, because it is kind of pneumatophore exit. It is can be easily traced, because this is one of the victim. This fellow is one of the victim, but whereas other two victims, these three personals are went. Other two victims went. Two are died. Only this personal dermatitis occurs na this format. Even he was somewhat rescued, because the sting is not tentacles is not, but in a huge way. So we made a survey and find out what is the jellyfish species exist. So in general, there are in some other Australian countries where exists an anti-venom for sting. Some box anti-venom exists for immediately they will be giving injection. So we also planned for a similar kind of injection with the package of practice with the medical doctors. So bringing out some of the facilities to having some kind of anti-venom. This is the species which are having giving this death. I'm sorry, it is not playing. So later it found it is like a box jellyfish, not it is not an hydrozoan. Okay. Similarly, we also create some kind of WhatsApp groups. It's like a initiation, because with the fisherman, wherever the sing kindly inform us. maybe we can give us as first aid measure, because we are not a full medical doctors, but initially as first aid measure, we can give some suggestion. Well, this is one of the divers who was doing diving in Tuticorin. So we initially told apply. It may not be the boxing. Some kind of outreach also we are doing for this kind of sting case studies. And similarly, and we are finding out some different jellyfish. First things, what are the diverse takes? There are many organizations are involved, but we are taking as a whole stretch all over the India. First, with what are the jellyfish species? What is the level of toxic, harmful species? What is the first aid measures? Like that, we are distributing through the fishes initially with the communities throughout the world, just throughout the country. Okay. Then some kind of kit also, because as I showed in the previous slide, in many of the countries like Australia, US, they developed some kind of kits to the fisherman. So we also developed certain kind of this kits and developed it to the fisherman, which these kits includes some pair of gloves, some vinegar, same of jelly card that for removing the clips. Okay, so and some local language will be giving some information to the fisherman. So how it will be. It is a initiation for this kind of first aid measures, and we are in the process of developing some kind of apps for the different jellyfish stings, whatever exists in our reports, and apps for the development of this. In future, it may be in takes time. In future, we'll be working on the similar line for bloom dynamics, everything. Okay, so that's all. But what I found is, because the marine idarians or it is an important resource, as I told. It's a huge resource. We're having different number of plants which having beautiful natures and fatality natures. However, even many students exist. The sense of latest biotechnology tools, people are not working on this basic science, because basic science which decides everything this biotechnology. So we have to group, we have to form some Nigerian groups and working on different resource. So the basic science, then coupled with the advanced molecular, because wherever now if you are going to a high impact journals, we have to go with some kind of advanced tools. So like molecular techniques has to be used, and then coming to the next level, because as it is having venom, and there are many toxic in India also, many venom studies and antibacterial or antiviral studies has been taken. But compared to other countries, we are in still this infancy stage. So we will promote some more research works on this Nidarians in all the aspects, starting from basic science, then the taxonomy, and then finally the anatomy, biology, molecular tools, then finally the next part, the drug discovery, this therapeutic potentials, everything. So as a whole, if you are telling, because Narayan is a very big resource, we come covering in 1 hour is very difficult. Even I also still in if you tell about the science, it is an infancy, because when we scrap any one species, we'll get a lot of information. Okay, so we will work on this in focus as in India and whole, because we all are all in India, and will interlink with many of the countries, how the global status. So all the Nigerians will be working. We'll hope for a better future on this Nigerian research. Thank you very much for our wonderful opportunities. If any doubts exist or any suggestions can be told. Thank you very much, madam, for the wonderful action. Thank you.
Dr. Ranjit, thank you so much. Thank you. Your presentation was vivid, going through the basics, detailed classification, and a very colorful presentation showing us all the Nidarians. and unraveling astonishing sea creatures. We are mostly aware of finfishes and shellfishes, but today we had a wonderful session on this. And moreover, usually research it is carried out in animal resources, viewing them as only potential resources for exploitation. But today we were inspired to appreciate and understand their biology, their distribution, and how basic research is very much important for carrying out sustainable environment and things like that. Thank you so much, sir. And you had also told us a current scenario of the Nidarians, how research is being carried out all over the world, especially focusing even on India. And you also shared your experiences. Thank you so much, sir. We
Okay.
were glad to have you with us today. We have few questions. So would you like to answer a few?
Yes, 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 Ramesh.
Okay. Sir, is there any edible jellyfish that is being processed near Tuticorin?
Yes, yes, yes. Previously, ma'am, actually, before 2005, there is some resource like Rofalima. It has been processed in Tuticorin region itself. Yeah, it has happened. But what the now fishermen are telling, we don't have any buyers for. We are also trying some, because in jellyfish is creating havoc and huge menace to the fishes, because we are working in a fishery institutes. So we'll also finding out. But as a whole in India, there is exists a fishery in Visakhapatnam. We are also reported. You can see a publication by Dr. Behra, who is working on who has published on the fishery of jellyfish in from Visakhapatnam region. There exists a good number of fishery. Even CMFRI, from CMFRI, there is a publication from Gujarat and Visakhapatnam. Even we have documented some extent in Kanyakumari, but and I export it. The fishery exists, but exporter has certain restriction with the government because the export certain kind of quality is required for that. So as of now, my answer is in Tuticorin as of is not there. It's because they don't have any much buyer for this export, but it exists before 2009. The particular place called Tarvekulam, they process the tentacles for making some kind of papad or something.
Okay, sir. And the toxic compounds which are usually secreted by these jellyfishes, are they
Yes.
normally hemotoxic or neurotoxic?
No, mostly they are neurotoxic, madam. Mostly they are.
Okay, okay. Do you know anything about the nutritional content of jellyfish, sir?
Yes, yes. Actually, my own scientist colleague is working on it. Mostly they have collagen and mostly the mucopolysaccharide. We are working on the line, ma'am, because as
Okay.
we are also starting in a couple of years back only. We are working on the nutritional aspects also, because
Okay.
the body as a whole, particularly the jellyfish, which having mostly the water content, other collagens and mucopolysaccharides exist, and you can see some of the publications on of this nutrition in future also.
Okay. Thank you, sir. Now, why do you think research is lacking in Nidarians in the recent
No, no,
years?
as a whole, I'm not lacking. I have not told, because
When compared to the recent molecular technology and protocols and procedures, compared, why do you think it is lacking? It is
no,
not
we
lacking, you say.
no what in general perspectives, because when science progress, two ways we can interpret. Okay, for example, when we see the Web of Science, I have taken data based upon the Web of Science database, where from 1998 to 2020. So two things exist. For example, some initial stage Indians, the publication level, which may not reach good impact factor journal. Maybe we restrict most of the publication within the Indian levels. Okay, which don't have that, but the work might be, we think it is very high, but the publication level point of view will be publishing in a low impact factor journal. That is one part. Now, apart from that, always the whole as a whole. We follow some of the most of the search as we follow protocol from the developed countries. We are not telling that it is in much lag. We are working, but our the output in terms of publication, it is not reaching up to the level of high impact journals. Maybe it can be refined by using some kind of, now people are going with the many tools, like that's why I shown some of the viewer. The representation form also creates an impact. And I think some of the Nigerians, like corals, we have good findings from MOEF, everything. So slowly, I think it will come in future also, ma'am. We'll hope for the best. I hope I answered in a direct way. Please.
Okay, okay, thank you, sir. And there is one more question from one of our professors. It is just a basic clarification. I'm working in an arts and science college. Is it
Yes, yes.
possible to carry out either basic or applied research? And even when doing a research, is it necessary to publish a paper?
No, it is depend upon the universities, ma'am. Sorry, because for
Okay.
example, in our university where I performed, you have to publish a journal, then only you will get awarded thesis.
Okay,
So that
okay.
is up to the institute level. But
Okay.
wherever, as I told, this publish, perish. So wherever certain information is there, you just put in a forum, because I also sometimes will feel when I found in the graduation level or PG level, sometimes we will see that we may not publish it. So unless if we publish, it will come to the open forum. So better, my suggestion is you do, you publish. Right?
Can you hear me, sir? Hello?
Yes, ma'am.
Ah,
Yes,
okay.
ma'am.
Okay. What is the scenario of corals pre-tsunami and post-tsunami period, especially in the southeastern coast of India?
Yes, madam, because there are many reports, but I am not worked much on the pre-tsunami and post-tsunami coral aspects. But there are many reports where it has been destructed, a huge diversion. Many universities has been, many institution, organization work on the post-tsunami aspects. There is because definitely these corals or hard corals particularly, it grows very, in a year it may grow, some of the species goes to 1 centimeter. So definitely whenever some events happens, the extreme event happens, definitely the coral resource get devastated. But as Indian government has rule, because if you see in many of southern parts of Tamil Nadu, even that buildings, late older buildings is made of corals only, coral stones. Now after the implementation of Wildlife Protection Act, there is a restriction of this natural picking or harvest of these corals. So such kind of thing, but extreme events definitely will have impact. But I have not worked on this post tsunami link.
Okay, okay, sir. Is there any ethical procedure to work on nidarians?
No, actually, as a whole, we can't tell, because definitely whenever we are working with some protected animals, we have to get permission from the concerned department, either forest or Minister of Environment. So that is there. And
Okay.
science, when we do any kind of drug related studies, yeah, definitely we'll follow some ethics, like animal model studies. If we are going, we have to get some consent from the concerned departments.
Okay, 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 sir. There is a question from a student. If we take jellyfish for our research, which study will be advanced and what are the techniques we can do with that, for example, in molecular biology?
Yeah, sure. Because in Indian condition, we are currently we are attempting on the taxonomy based on molecular tools. We
Okay.
have some good amount of DNAs and DNAs of many species. So coming to this studies, uh, starting from scratch itself, because I go with basics first,
Okay.
because you see what is the animal, how it looks, you get the feel, then only we can go. Because molecular, it is like 1 gram or something less than that only we'll be taking. You couple with molecular tools, with if you are concerned with the degree, nature of degree, UG or PG or PhD, there are many works can be done. Even
Okay.
if you tell jellyfish taxonomy, molecular taxonomy, it is a resource. And if you are working in venoms, one species venom itself a resource. If you find in ecology, one species itself a resource. So there are the humpty number of you can explore, because that's why as a whole I told there are humpty number of options to work on, because even the ecology aspects, there are Susan-Late jellyfish, Susan-Late corals. So there are many works, many works which is published in Nature's and many high impact journal. So definitely you can work in any specific title if you require through madam you can send, we will suggest in this regard.
One last question, sir. Thank you. A person has said that he has seen in a video a man applied urine to reduce the pain.
Yes, it is there, because the sting management, there is a big books. We are also having reproducing some kind of documents.
Okay.
Yeah, it definitely reduces the sting. Okay. And there are many reports. Other report also you can use cardiac oil and some kind of turtles. Many people turtle oil for defense. So there exists the video. I also seen that videos. So we can use it, but trial form you should not use this for all the species. You should know what species what should be applied. For example, vinegar can be applied in for one species, but it may not be applicable for other species. Similarly, hot work can be applicable for one species, cannot be applicable to that. To know the knowledge of the people, we have to use. One more thing I am telling, because if you see the fisherman, because we are dealing with the fisherman, when we go there, some of the bugs which they simply take it in hand, because when we use our hand, the palm area, that white portion of the area, the sting cannot penetrate as much, rather in the other side. So they simply take the box jellyfish in hand and throw it. No, it is up. There are many videos showing that, and some of the videos also they just put the sting and what happens after that. Like that, many things that happens, but the usage should be correct. We have to develop some package of practice for different species. What is the application as a first aid? Because as a other thing, it will come in the second part. We have to do some package. We are also developed, and some of the things that has been displayed in our pro slide. Next.
Okay. Thank you, sir. Thank you so much. I think you've answered almost all the questions from the participants. We thank you so much for your valuable presence today with us. Though you were busy with many works, you consented to be with us today, and we appreciate it. And on behalf of the organizing team, especially the Department of Zoology from St. Xavier's College, we extend a warm thanks from the bottom of our hearts, sir. Thank you so much. Thank you,
madam. I also thank you was your institute, your college, for inviting me for this event, for delivering lecture. And in this occasion, I used to thank our director of our institute, who permitted me to present this topic. And similarly, the PIs of like PAs of jellyfish project, because we worked on that also. And then some of our we are usage of NAM, the usage of our Vivo server and Web of Science from ICR as a whole, ICR and CMFRA. I have to thank for delivering the lecture, and I hope act on in future. And if anything will be rectified, there are many things from our projects we have found out, but I framed, because it's a wide group of audience, we have to have some basic things, gets insight on it, and what are the advanced things, what is the current status, like that event. So if anything, you can ask madam, or you can email me. Thank you very much for this opportunity. Thank you, and a very good afternoon to everybody.
Thank you so much, sir. Thank you. Now I request Dr. Elizabeth Tangamani Sunitha, Assistant Professor in the Department of Zoology and the Organizing Secretary of this National Level Zoology Webinar Series 2020, to propose the formal vote of thanks.
Good afternoon all. I am Dr. T. Elizabeth Tangamani Sunika, one of the organizing secretary of this webinar. Saying thank you is more than good manners. It is good spirituality. On behalf of the management, I will extend a very hearty vote of thanks to our resource person, Dr. J. Webin Prabhudas and Dr. L. Ranjit, for gracing your work and sharing with us your findings and opinions today. We are thankful to our respected Reverend for the Rector, Reverend for the Secretary, Reverend for the Principal for the motivation and enthusiastic support. Our special thanks to the organizing committee, teaching and non-teaching staff, Department of Zoology for the continuous support and coordination. I also extend my sincere thanks to Dr. Luduswamy, IQSC coordinator and IQSC team for making this webinar a great success. I would also like to thank our head of the department, Dr. P. Raja, for his moral support and guidance. And as a co-organizing secretary, and again, I thank Dr. S. Mabel Parimala making this webinar a great success. I would also like to thank Dr. Winston, Assistant Professor, Department of Computer Science, for his technical support in this webinar. I express my sincere thanks to all the participants. Your participation is additional strength to us. Thank you all for your kind participation. With these warm words and a kind message, we move to the end of today's webinar. I thank all for your cordial cooperation. Kindly join us with us in the future endeavors. Achieve big in life, always dream big and work hard to get there. Thank you, one and all.
Thank you. Thank you.
Thank you all. We have posted the feedback link both in the Zoom platform as well as in the YouTube live chat. Kindly submit the feedback form. It will be open only for 15 minutes. And once you have filled the feedback form, you can leave the meeting. Thank you so much for your participation.
Sam? Sam?